What we have seen here at Lake Mead compared to what we found in '07 is that we have our clarity has increased about 13% kind of overall the whole Boulder Basin and the whole Lake Mead area. And we also have seen a decrease in the chlorophyll counts, about 45% decrease just in the Boulder Basin. Of the other areas of the lake, we haven't seen it decrease as much, but we also have a lot of quagga mussels in those basins too, so we're not sure if it's the quagga mussels or something else that's causing the decrease in the chlorophyll. So we also, with the decrease in the chlorophyll, that's usually mostly associated with the free-floating algae. So we've actually seen an increase in filamentous algae, mostly near the dock structures. They're attaching to the docks and to the buoys and other structures that we have in the lake. And we have 3 species of the filamentous algae. So, and it's thriving more than it ever has before out here at Lake Mead. So we're not sure if the quagga mussels are filtering out the free-floating algae enough, which is competition for the filamentous algae. So the filamentous algae is actually increasing because it doesn't have as much competition. Most of our plankton tows that we've done, we started those prior to 2007, and also we have other agencies that are testing the water and doing plankton tows for many, many years. Lake Mead is probably the most tested water, in the whole country with different agencies because it's a drinking water source for 20 million people. And so what we have found so far is during peak breeding times of the year, or the peak spawning times, the veligers of the quagga mussels make up 80% of the biomass of some of our samples, our plankton tow samples. So they're kind of displacing rotifers and copepods and other organisms, but then it goes back down to where everything's fairly even during other times of the year. And that 80% is just in a few samples. It's not all lake-wide, so it fluctuates depending on where you're taking the sample on Lake Mead. So we still can't say whether it's affecting the ecosystem or not. For the fish don't seem to care whether the biomass is made up of copepods or veligers from the quagga mussels. There's still food source there. And we have not seen a decline at all in the fish. We've actually seen an increase in the fish populations. Yeah, what we have found in the mussels at Lake Mead is that their growth rates, they grow pretty fast here, and the smaller mussels grow faster than the larger mussels. So they kind of reach a apex and then they kind of start growing slowly. And also what we've noticed is that our mussels they grow throughout the winter, which is unheard of in any other place that they're located, according to the data that I've done some research on and some other researchers have done. So we haven't ever found any place that any evidence that they are growing in the winter, so that's pretty unique to hear. So we know that they're active all year long, we know they're filter feeding all year long, and they're also reproducing all year long. According to our veliger counts, we get counts all year long, but they do fluctuate. We get higher veliger counts in the early spring and then in the fall. And the growth rates that we have of the smaller mussels, they grow 0.05 millimeters a day, which is actually pretty quick, pretty large growth rate. The depths that we have been finding the mussels at, we've only gone down to 400 feet because that's as far as we can get with our ROV, and I have found them at 400 feet. The lake's another probably 150 feet deeper than that. They are in very low concentrations, and they don't have a lot of pigment in their shells, so they're pretty much white. And we don't know if they're reproducing at those depths. They might not be reproducing, but that's still another study that we have to do, or we'd like to do. The densities that we have found from 2007 to 2009 is our mussel densities went from about 500 per square meter up to 9,000 per square meter in the hard substrate areas, just in the Boulder Basin. And in the soft substrates, they went from 8 individuals to 3,000 individuals per square meter. But we also have areas around Lake Mead that are outside the Boulder Basin that we have concentrations up to 55,000 per square meter. And that area where we have 55,000 per square meter is 33 miles away from Boulder Basin and where the initial infestation was. And that increase from 0 to 55,000 was in less than 1 year. And it was 33 miles up the lake, upstream from the original infestation. Yeah, with the number of mussels, and we're not sure if they are increasing. I mean, they were increasing from 2007 to 2009, and we're still analyzing all the data from 2010 that we've collected. So we're not sure if they're increasing. It seems like they're running out of places to inhabit. They've got, if you look at some of the rock samples from underwater, the mussels just keep stacking on top of each other and on top of each other. So they're running out of places to attach, except for attaching to other mussels. So we won't know for sure until we finish all of our data, finish processing all the data. The spawning cycles for the mussels, we know that there are two peaks, the peak in the spring, early spring, and then a peak in the fall. But we still have veligers all year long in samples, so we assume that they're breeding either continuously or multiple times during the year, more than they would back east. And with our evidence that we know that they're growing even in the winter, they're still actively feeding, and so they're probably still spawning, but we don't know for sure. We haven't collected mussels all year long and checked them to see if they're reproducing. I anticipated, if we got mussels, I didn't anticipate the problem would be this big. I knew it would be a major inconvenience and it would be a cost expense to the park. But just the rate that they took over the lake, we had small populations. They're well established, but they're small populations. But they just exploded in 1 year, just from one basin clear to all of Lake Mead and all of Lake Mojave. And they just wreak havoc on any kind of an intake system or a pipe system or boats. So anything that's in the water that they can attach to, they'll attach to. And we have buoys that sink. We have to maintain all of the aids to navigation buoys, and every dock structure, every boat, every line, every piece of rope in the water that they colonize, everything. And all the beaches now, as the water levels are dropping, the beaches are getting mussels exposed and they're dying, and so people are walking on the beaches and cutting their feet. And so we haven't seen it as bad as the Great Lakes, where you have huge piles of dead mussels washing up on the beaches. I'm hoping we never get there, but that'll be another huge problem that we'll have to figure out how to handle once it happens. Yeah, another issue that we're having here at Lake Mead is we have quite a few submerged cultural resources. And we have a B-29 bomber, we have a concrete aggregate plant that was used to build Hoover Dam. That's all underwater, and some other wooden boats and some other structures underwater. And all those are getting covered up with quagga mussels. And we have some old cyanide tanks that were used for mining, and all the walls are collapsing on these tanks just because of the weight of the mussels. And also on the B-29, certain areas of the wings are covered in fabric instead of metal. And with the whole plane is just totally covered in mussels. And the weight of the mussels is causing that fabric to crush in, and it's just deteriorating the plane overall. And it's been in the water for many years, and it wasn't deteriorated hardly at all until the mussels just colonized it and just covered the whole thing up. And it's also causing the metal to break down quicker. We've had some substrate samples that we put in the lake that had the same metal material that was used to make the plane. And what we do is we periodically pull them up and scrape all the mussels off of it and look at the substrate. And the mussels, when they attach with their byssal threads and the glue that they use to attach to the byssal, to attach to the substrate, is causing the metal to dissolve. So it's got all these pock marks in it and stuff. So we're going to lose all of our cultural resources a lot quicker because of the colonization of the quagga mussels. Yeah, it's very important, if you do not have any mussels in your lake, to come up with a program that you inspect boats that come into your lake, and a boat can't launch on your lake without getting inspected. And I know a lot of states are doing that currently. Depending on the size of your lake, it's some lakes that might not be that feasible, or if you're open 24 hours a day. I know a lot of the state lakes are only open during business hours, and they can close the gates at night and not allow people to launch, and they can also close their launch ramps during areas when there are nobody there to inspect. But the headaches of getting quagga mussels and just the economic impacts and the ecological impacts way outweigh the cost of prevention, of keeping the mussels out before they get there, and any other invasive species that are carried on boats.